Hello everyone, welcome back. I'm here with the genetics part 2. Uh, in our last class, we learned about genetics, what is genetics, and the terminology related to it. For example, heredity, the definition of genetics, gene, alleles, etc. Now, today we will learn the next part of the chapter, that is Mendel's experiment on inheritance. Okay, so first let us talk about genetics. We have already learned about the genetics. Genetics is the study of transmission of genes from parents to their offsprings. Okay. Now, Gregor Johann Mendel is also known as the father of the genetics. I had asked you question in my next class in my previous class, that why Gregor Johann Mendel is known as father of genetics. You may have done the assignment, or you may have done the work. Now continue with it. Gregor Johann Mendel conducted breeding experiment on garden pea, Pygium sativum, out of sheer interest. Now he was not a biologist; he was an Austrian monk who used to teach mathematics. But he was a very enthusiast person or interested person in biology. So what he did is that in his interest, he performed breeding experiment on garden pea, whose scientific name is Pygium sativum. Now see here the scientific name of pea, that is Pygium sativum. I have underlined it because it is the scientific name, and in scientific name the first is the generic name, and another one is specific epithet. Generic name should always start from capital letter, and specific epithet will start from the small letter. So Pygium sativum of Pygium sativum. Now Mendel took various contrasting pair of characters of P and performed cross. That is, cross means monohybrid cross or the dihybrid cross, taking those pair of contrasting characters. Now what is monohybrid and dihybrid we will learn later in the same class. So he took one dominant and one recessive character. So in those pair of characters, one used to be dominant and another one used to be recessive. Now what is dominant, what is recessive, I have already explained you in my last video class. So dominant and one recessive character within those pair of contrasting characters. So in those pair of contrasting characters, he took one dominant and one recessive character. For example, if you see in case of Pygium sativum, he took one dominant and another one as recessive character. So flower color, in flower color, dominant one is purple color and recessive one is white color. So likewise, there are number of characteristics which he took as one is dominant and another one is recessive. Example, in this case also, in seed color, dominant is yellow and recessive is green. Seed shape, round is dominant, whereas wrinkled is recessive. So pod color, green is dominant, yellow is recessive. Pod shape, inflated is dominant, constricted is recessive. Flower position, axillary. Axillary means arising from the nodes, and terminal means arising at the top of the shoot. Okay. Plant height, tall can be termed as dominant, and he took dwarf as recessive. Now, taking all these characters, he performed monohybrid and dihybrid cross. Monohybrid cross. Now, moving on to the monohybrid cross. What is monohybrid cross? The cross performed between the two plants using or taking single pair of contrasting characters. That means if I took single pair of contrasting characters from this one, for example, if I take only seed color, for example, seed color is yellow and green. If I take only seed color and perform the experiment or crossed between the two plants, that type of cross is known as monohybrid cross. So in this case, the cross performed between the two plants using or taking single pair of contrasting characters. For example, cross between terminal and axillary flower position. So those type of cross is known as monohybrid cross. Now you can clearly see one is dominant character and another one is recessive. Dominant character is axial means axillary flowering, and recessive means terminal, in which the flower takes place at the apex of the shoot or top of the shoot. Now parents Capital AA can be termed as the gene or genotype of the axial flower, and small a can be termed as the genotype of the terminal flower. Now, what is genotype and what is phenotype? I've already explained you in the previous class. You need to, if you don't understand again, if you don't know genotype and phenotype, please refer the previous video class in which I have discussed about the terminology related to the genetics. So axial is axial. The genotype of axial is AA, or the gene of the axial flowering is AA, capital A, whereas the gene of terminal flowering is small a and small a. Now there are two different plants, one bearing axial flowering and another bearing terminal flowering. So they the cross occurs, or the cross is carried between these two different plants, one bearing axial flowering and another bearing terminal flowering. So those those cross between the two different plants are termed as cross pollination. So here the cross pollination takes place, and as a result we know during the gamete formation only one of the gene from the pair takes participates. So one a capital A from one axial bud, and small a from terminal flowering. So in F1 hybrid we see that both are the combination of both the axial as well as terminal. But since axial is dominant, and therefore this can be termed as this plant bears axial flowering. This plant bears axial flowering. Okay, so in F1 hybrid the plant bears axial flowering. The cross carried out in between the axial flowering plant and terminal flowering plant, as a result you will get F1 hybrid as axial flowering plant. Okay, with the gene of both the axial flowering as well as the terminal flowering. But since axial flowering is dominant, therefore the character shown by the plant will be axial flowering. Now the back cross or the second cross between F1 hybrid plants take place. So F1 hybrid, this is the gene of F1 hybrid, and this is also the gene of another F1 hybrid. So capital A small a we got as a result of the cross between the axial pure axial plant and the axial flower bearing plant and pure terminal flower bearing plant. We got the hybrid as capital A and small a. Now this capital A and small a, that is F1 hybrid plant, we did a cross between the two F1 two plant that bears, that is from the F1 generation, that is capital A small a, capital A small a. As a result we produced four plants. Okay, we produce four plants. Now this is in case of self pollination. We are not talking about the cross pollination, but it is self pollination. Okay, now this capital A and small a, it produces two type of gametes, capital A small a, and this also produces two type of gametes, capital A and small a. Now the combination can be of four types. You can see here capital A, this A and this A combines together to form capital A, this A and this A combine together to form capital A and small a, this small a and capital A combine together to form capital A and small a, and this A as well as this A combine together to form two small a's. Thus, if you see the phenotypic ratio, phenotype means the expressive characters which are giving the expression, which are expressing itself. So axillary flowering, since capital A is dominant and it is the character of axial flowering, therefore there are three axial flowering in this case. So phenotypic ratio will be axillary is to terminal. So there are three, 1 2 3 axillary flower bearing plant, and there will be one terminal flower bearing plant. So the phenotypic ratio in case of monohybrid cross is always 3:1, and in case of genotypic ratio it is capital A, that means pure axillary flower bearing plant. That is next one is capital A small a, that means hybrid in between both, and another one is pure terminal flower bearing plant. So the ratio is 1:2:1. So thus the genotypic ratio in monohybrid cross is always 1:2:1. Now if you don't understand, I have made a checkerboard over here. Capital A and small a from this one, and capital A and small a from this one. So both of the plant bears two type of gametes, capital A, small a, capital A and small a. Now if you do the cross between those gametes, capital A, this capital A and this small a, this small a and this capital A, and this will be giving you this one. So that is the ratio. Genotypic ratio will be 1:2:1, and phenotypic ratio will be 3:1. Now let us see if it is correct in case of other characteristics also. Now as I already stated, in monohybrid cross you take only one contrasting character. In this case the contrasting character is for axillary flowering and terminal flowering. Now in next case I will take the character of the pure tall plant and the dwarf plant. So monohybrid cross between tall and dwarf plant. So tall T plant is represented by the gene capital T, whereas tall dwarf P plant is represented by the gene small t. Now parents, one is pure tall capital T, and one is pure dwarf small t. Now both of the parents contribute one gamete from one gene in his gamete to form capital T small t in F1 generation. Now this one is hybrid tall. This is hybrid tall. Nor it is pure tall, nor it is pure dwarf, but it is a hybrid tall. But since capital T is the dominant gene over here, so we take it as the as the tall plant. Now again we have to do the cross between the F1 hybrid plant. So F1 hybrid 1 capital T small t, F1 hybrid plant capital T small t in the same parents. This is in case of self-pollination. Now you can see this capital T, this plant produces two type of gametes, capital T small t, and this also produces two type of gametes, capital T small t. When you do the cross between T, you will get capital T small t, capital T small t, and this both of them small t. So these are the four type of plants produced in F2 generation. This is F2 generation. After F1 there is F2 generation. So in F2 generation there are three tall plants and one dwarf plant. So phenotypic ratio in this case is also tall is to dwarf, that is 3:1, and genotypic ratio, that is tall is to hybrid is to dwarf, that is TT capital T small t, that will be reached up to 1:2:1. So here is the checkered board. Okay, so capital T small t, capital T small t, and small t. Okay, so this was about the monohybrid cross. Now what are the principles based on above monohybrid cross? So what we learned. Now each pair of contrasting characters depends on a pair of genes. Now pair of contrasting characters depends upon a pair of genes. As I already told you, in monohybrid cross we have seen that this is a contrasting character, that means pair of gene TT, that is pure tall, and TT is this pure dwarf. They depend upon the pair of genes. This is a pair of gene. Okay, if both of them are there, then the plant will restore its own original size, that is original height. In case of this one, you can see capital T and capital T, it is a pure tall, and small t is a pure dwarf. But in this case capital T and small t, so it becomes hybrid tall. It is neither tall nor dwarf. So in this case the gene loses its original height. Okay, but it comes to the hybrid tall. Thus the pair of contrasting pair of contrasting characters depends on a pair of genes. Okay, now next one, each and individual carries such genes in duplicate. I already told you there are two genes, one duplicate for another one. Okay, so each pair of genes, each individual carries such genes in duplicate. An individual produces gametes which have only one member of pair of genes. So again coming back to this one, you can see in F1 generation this contributes one gamete and this contributes one gene. This contributes one gene, that is capital T. This contributes one gene, that is small t. So that means an individual produces gametes which have only one member of pair of genes. Fertilization of the gametes restore the duplicate condition of the genes. Now here you can see there is a fertilization between two type of gametes. This is one gamete, another gamete is this one. So when the fertilization takes place, that means they combine with one another. Thus the pair of the gene is again restored. Okay. Fertilization of the gametes restore the duplicate condition of the genes. Next one is sex cells. With respect to the genes, they contain get fertilized at random. Now in this case, sex cells, they can fertilize at random. It does not, this T does not require capital T to get fertilized. It may get fertilized with a small t also. So thus the fertilization is random in case of monohybrid cross or even in dihybrid cross. Okay, so that was about the monohybrid cross. Okay, now moving on to the dihybrid cross. So di means two, so taking the two characters. So it is a cross between, definition goes like this: it is a cross between the two plants taking two different pair of contrasting characters. Now it is cross between two plants taking how many how many pair of contrasting characters? Two different pair of contrasting characters. For example, in this case, we are in dihybrid cross, we are doing with seed color and seed shape. So these are two different characters, and the contrasting character of seed color is yellow and green, and seed shape is round as well as wrinkled. Now round and yellow both are dominant gene, whereas wrinkled and green both are recessive gene. So we are taking the two pair of contrasting characters, two different pair of contrasting characters. So round and green is one pair of contrasting characters, round and wrinkled is one pair of contrasting characters, yellow and green is another pair of contrasting characters. So that means two pair of different contrasting characters. One is for color and another one is for shape. So thus is known as dihybrid cross. So parents cross, so pure parents cross. Parents cross is always pure. So this one is R capital Y. We know that each of the gene has its own duplicate. So this R, that is round, it has got duplicate gene, another duplicate gene, that is capital R. It has also got another duplicate gene, that is capital Y. Similarly, in this case also they have duplicate genes. Okay, so RRYY, that means this one is for green, round and yellow. So the shape of the seed is red, round, and the color of the seed is yellow. In this case, it is wrinkled, 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 and green. So in this case, in another parent, the shape of the seed is wrinkled and the color of the seed is green. So we start a cross between the round yellow seed and the plant which bears round yellow seed and the plant which bears wrinkled green seed. Now this is a whole gene. Now it produces gametes. This plant produces gametes which contains one capital R and one capital Y. Similarly, this also produces gamete which contains single gene of character. So this from both R, one R is present, and from both Y, one Y is present. Now when the fertilization occurs, takes place, fertilization. Okay, so when fertilization takes place, they form the new pair of gene, that is F1. In F1 generation, you can see they contain both the characteristics from round yellow as well as wrinkled green. So this is in case of F1 generation. So it is a hybrid. It is a hybrid, that means from both the cases. Now moving to the F2 generation. Now again I will do the cross between the both. So from each parent, 4 gametes are produced. Now in next cross, where I will perform self-pollination between the F1 hybrid RRYY, it will produce 4 type of gametes. 4 gametes are produced from each parent. From this parent also 4 gametes, and from this parents also 4 gametes. So this is in case of F1 generation. Okay, now this will go to the F2 generation. Now this R and this Y make a pairs of and form a single gamete, that is capital R capital Y. Similarly, it will produce another gamete, that is capital R small r capital Y, and small r small y. How it is produced? Capital R small, capital Y small y, they cross with one another. Capital R capital Y, capital R small y, small r capital Y, small r small y. So this is how the four type of gametes are produced. Similarly, in this case also there are four type of gametes for ry, 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 and small r small y. So the four type of gametes are produced. Now these are the gametes, these are the gametes which we will which will combine in the F2 generation. F2 generation. Okay, now moving on to the cross. Okay, I've already made here. So capital R small r capital Y small y, capital R capital small r capital Y small y. This is F1 generation plants. F1 generation. Now when F1 generation into F1 generation, so this is self pollination. It is not cross pollination, but it is self pollination. Self pollination. I hope you know the self-pollination or cross-pollination. If you don't know, you can mention in the comment. If you need more explanation regarding self-pollination, you can always mention in the comment. And if you don't understand any points that I have explained, you can also mention those things in the comment, so I can explain you again. Okay, so in this case, all this particular plant produces four type of gametes. I have written in vertical columns, and this another plant produces four gametes. I have written in rows. Okay, now when the cross between two takes place, this one here, capital R capital R capital Y capital Y. Now again, capital R capital R capital Y small y, this and this, capital R small r capital Y, and capital R small r capital Y and small y. Okay, now this one, capital R capital Y small y, this one capital R small y. Now this one, capital R small r capital Y small y, and in this case capital R small y small y. Again coming back, this is capital R small r capital Y. In this case capital R small r capital Y small y. In this case small r capital Y. In this case small r capital Y small y. In this case capital R small r capital Y small y. In this case capital R small y. In this case small r capital Y small y. And in this case small r small y. So in this case we can find the ratio. So we know ry is the dominant gene. Okay, so would the gametes the zygotes, or the after fertilization, those bearing capital R and capital Y, those are the dominant gene. That means they are round and yellow. Okay, so I have written it round and yellow, capital R and capital Y. So let us count how many are there in case which bears capital R and capital Y. So in this case capital R capital 1, this is done 1. In this case also you can see capital R and capital Y, so this one is 2, done. In this case also you can see capital R capital Y, this is third one. In this case also you can see capital R capital Y, fourth one. In this case also you can see capital R capital Y, 5. There is no capital Y over here, but in this case you can see capital R capital Y, so this one is 6. In this case there is no capital Y. In this case there is capital Y capital R, that this one becomes 7. Capital R capital Y, 8. And finally capital R capital Y, it becomes 9. So those bearing capital R and capital Y, there are how many plants are produced? In case of 16, 9. Okay, now let us go with capital R and small y. Capital R, small y means round and green. So capital R, those which are taken already, now leaving up, these are capital R, small y. Okay, so this one becomes 1, capital R, small y, this becomes 2, and capital R, small y becomes, this becomes 3. So capital R, small y, it contains three plants which whose seed will be round and green. Now small r, capital Y, that means wrinkled and yellow. So small r, small capital Y, so this will be 1. So I am writing A so you can understand. Small r, small capital Y, that means another one. Small r, capital Y, that means another one. So 1, 2, 3. So three plant bears wrinkled yellow seed, and only one plant bear pure wrinkled and green seed. So total there are 16 plants: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Out of 16, 9 plant bears round yellow seed, 3 plant bear round green seed, 3 plant bears wrinkled yellow seed, and 1 plant bear wrinkled green seed. Since it is a expressive, it is expressing itself, therefore it is known as phenotype, and the phenotypic ratio of any of the dihybrid cross is 9:3:3:1. Okay, so this was about the dihybrid cross. Hope you have understood. If you have any problem, you can always mention in the comment. I will again discuss this chapter. Now moving on to the next part, that is law of inheritance, Mendel's law of inheritance. After performing such series of experiment, such series of experiment, that is monohybrid cross, dihybrid cross, Mendel proposed 3 laws. Okay, or Mendel's, it is also known as Mendel's law of inheritance. Number 1, law of dominance. Out of a pair of contrasting characters present together, only one is able to express itself while the other remains suppressed. In this case, you can easily see, I have told you, in this case, that is let me take the example of monohybrid cross, and that will help you more. Okay, so out of pair of contrasting characters present together, you can see here capital T and small t, both the contrasting characters are present together. That is, capital T is for tall and small t is for dwarf. Out of pair of contrasting characters, okay, that is capital T, small t, only one is able to express itself while the other remains suppressed. Only one is able to express itself and the other one is suppressed. For example, among TT, we write it as tall, or we write it as hybrid tall. In such case, what happened is that T, capital T, it expressed itself. It expressed itself, while small t, it became recessive, or it gets suppressed. Okay, so since both the genes were present, both the genes for tall as well as dwarf was present, but only the tall was able to express itself, so thus it became dominant over the dwarf, over the gene of the dwarf. Okay, so that is the reason the law of dominance states that out of a pair of contrasting characters, both of a pair of contrasting characters present together, only one is able to express itself while the other remains suppressed. In such case, T is capital T, that is the gene of tall was able to express itself, whereas the gene of dwarf, it became suppressed. Okay, so that was the first law, the statement of first law of Mendel's law of inheritance, law of dominance. Now moving on to the next part, that is law of segregation, also called the law of purity of gametes. The two members of a pair of factors separate during the formation of gamete. As I already told you, if you take out the TT, that is hybrid tall, when you start a cross between capital T and small t, capital T, small t, two hybrid plants, then obviously they will separate, they will form the individual gamete over here. they are forming the individual gamete. Okay, so the two members of a pair of factors separate during the formation. So although they are the gamete, they are the they are the character gene of the same gene of the characters from the same plant, but when during the formation of gametes they are separating with one another. Okay, it will act as individual gamete and this will act as individual gamete. So in this case they get separated. So thus the law of segregation states that the two members of a pair of factors separate. The two members of a pair of factors separate. They are separating during the formation of gamete. So this is the gamete. So during the formation of gamete, both of them separate with one another and act as individual gametes. So next one is law of independent assortment. When there are two pair of contrasting characters, the distribution of the members of one pair into the gamete is independent of the distribution of the other pair. For example, if you say if you see the dihybrid cross, so they have stated that when there are two pair of contrasting characters, one is round, wrinkled, yellow and green. So two pair of contrasting characters, the distribution of the members of one pair into the gamete is independent. So in this case also you can see it forms the four type of gametes. One is capital R, capital Y, and a capital R, small r, capital Y, and a small r, small y. But it is not certain that R, that is round, should be pairing with of the yellow. It is not mandatory. So they are independent of one another. Round, it can pair off with yellow and it can pair off with the green also. Similarly, yellow, it does not need to pair off with round. It can pair off with the wrinkled also. So that means round is round the round character is independent of yellow character yellow character and vice versa. Okay, so round is not dependent upon yellow. That means when they pair up, it is not mandatory that round should pair up with yellow. It can pair up with green also. Okay, forming a pure gamete. So this is what the law of independent assortment tells us. When there are two pair of contrasting characters, you can see this is round green, yellow, round wrinkled, yellow green. Contrasting characters, the members of one pair into the gamete is the distribution of the member of one pair. Is independent of the distribution of the other pair. So it is not mandatory that a round should pair off with yellow. It can pair off with the green also. So that is what the law of independent assortment states. Finally, the last part of the chapter is upon the mutation. Now mutation, what is mutation? Mutation is a sudden change in one or more genes or in the number or in the structure of chromosomes. So the number of chromosomes can be altered, the structure of chromosome can change, or there can be change in genes. So those type of sudden changes are termed as mutation. And excuse me, I'm so sorry. Mutation mutation are temporary as well as permanent. Most of the mutation are permanent, while some mutation it may be altered to the previous one. So some of the mutation there are some diseases caused by the mutation. So one of the diseases is sickle cell anemia. And what is sickle cell anemia? It is a blood disease caused by the gene mutation. So when there is a gene mutation, there is certain gene mutation takes place, and person can suffer from sickle cell anemia. So it is it is a mutation of DNA which controls the formation of formation of RBCs. So when there is a mutation of DNA which controls the formation of RBCs, at that time the sickle cell anemia takes place. Now we have already known that during World War II there was atomic bomb blast in Hiroshima and Nagasaki, and we know Hiroshima and Nagasaki, that is in Japan. It still there are some people, still there are some people with lot of deformities. So there are plants with a lot of deformities, there are animals with lot of deformities. How it took place? Because the radioactive radiation, they can also they can also alter the gene structure. Alter means change the gene structure, and when you change the gene structure, it is permanent. Thus it will continue generation after generation, generation after generation. Thus the mutation can occur from radioactive radiation, and it is persistent and permanent in case of every living organism. We also know that there are some deformities we have seen in social medias, where there are people with two hairs, there are people with six fingers, there are people with arms coming out through their chest. What is why all this happened, all these deformities took place? Because of the change of mutation. For example, the gene of your ear is AAA. The gene of your ear is AAA, and the gene of your head is AAG. Okay, so for example, somehow what happened is that due to the radioactive rays, or you were exposed to the radioactive radiation, this A somehow get deleted, and in place of A, G came over here. That means in place of your ear there will be the formation of head. So this is how the people with two head are born. Okay, so this is because of the mutation. Now this finally wraps up our chapter number 2, that is genetics. Now we will continue with the another chapter in our next class. So stay safe, take care, stay at home, don't go outside, and if you have any problem, please mention in the comment section below, as well as share and subscribe for more updates. Thank you so much.